In diagnostics, biosecurity, and environmental science, one question can change everything. How do we find a microbe's unique DNA fingerprint? That's why we built iSignify, a lightweight, offline-first AI application that identifies unique microbial DNA signatures in minutes, not weeks. iSignify uses a fast, on-the-fly K-mer comparison engine to identify signature regions found only in your target genome. It automatically pre-processes messy genome files, merges contexts, and aligns background genomes, all locally on your device. Then Google's Gemma model steps in to explain the results, translating raw numbers into clear summaries you can actually understand. Researchers can now create diagnostic tests for emerging pathogens in the field, monitor water systems for pollution, or track engineered microbes with synthetic DNA barcodes, all without sharing sensitive data, or even needing the internet. Here's iSignify in action. Upload your files, run the analysis, and get a downloadable list of unique signatures, ready for PCR primer design, database indexing, or real-time threat detection. With iSignify and Gemma, we made microbial fingerprinting fast, local, and accessible. This is what science looks like when it's in everyone's hands. iSignify, built for the Gemma 3N Impact Challenge.